The Earth's geologic history is divided into 4 eons, that being the Hadean, Archean, Proterozoic, and Phanerozoic. In terms of the origin and evolution of life's metabolic systems, the data we have suggest that much of it occurred throughout the Archean, which spanned from 4 to 2.5 billion years ago. The reason the start of the Archean is set at 4 billion years ago is because that's the age of the Acosta gneiss, the oldest rock we have yet found. This eon began around the same time simple single-celled organisms emerged from self-replicating reactions in the environment, and ended with carbon being cycled around oxygen in a manner similar to today. The Archean itself is subdivided into 4 eras, that being the Eoarchean, Paleoarchean, Mesoarchean, and Neoarchean, and these can be described by the element which served as the electron donor to drive primary productivity at the time. If you want to know more about how these metabolic systems themselves evolved from pre-existing ones, you can watch my videos, What Was the First Organism Like?, From the First Cell to Luca, The Origin of Photosynthesis and Respiration, and The Origin of Aerobic Metabolism. The first, the Eoarchean, spanned from 4 to 3.6 billion years ago and can be described as the Hydrogen Age. The first organisms likely lived in a hydrothermal system about 4 billion years ago. When we think of a hydrothermal system, we often think of a deep-sea black smoker. This kind is powered by interactions between pore water in the rocks and magma, which superheats it, forming a black smoker plume. However, there are other types of hydrothermal systems, most notably serpentinizing systems. Here, water chemically reacts with ultramafic rocks such as olivine and pyroxene to form serpentine, magnetite, and molecular hydrogen. At the beginning of the Archean, the crust was still chemically undifferentiated from the mantle, meaning ultramafic rocks were far more common on the surface than they are today. As such, these would have been found on both the ocean floor and on land. I should note that at this point, land didn't exist in the form of continents, and instead as small islands in a vast global ocean. However, exactly how large these islands could get isn't well constrained. When studying prebiotic chemistry, one thing that stands out is the importance of hot-cold, wet-dry, and acidic-basic cycles to drive it. These would not have occurred in marine serpentinizing systems, suggesting the cradle of life was a hydrothermal pool. In serpentinizing systems, hydrogen will react with carbonates to form organic molecules. These reactions are catalyzed by various minerals in the environment, and early organisms could have utilized these to grow. At this point, the metabolic processes in the first organisms would have been largely, but not entirely, driven by processes we'd identify as abiotic, and, since they would be occurring within the confines of the hydrothermal system, at this point, life couldn't venture out from its cradle. Some reactions would be driven by biocatalysts such as ribozymes, but like all reactions, they have to be thermodynamically favorable to proceed. Modern organisms use the hydrolysis of the phosphate group on an ATP molecule, binding an organophosphate group to a biomolecule, thus activating it. After the molecule goes through the reaction sequence, the organophosphate group is released as an orthophosphate. The ATP is regenerated by reacting an ADP with an orthophosphate, a reaction which requires an input of energy and complex molecular machinery. Molecular machinery that the first organisms wouldn't have had. For a long time, astrobiologists were at a loss as to how the first organism powered its energy metabolism. However, the answer may lie in the heavens. There's an iron-nickel phosphide mineral known as Schreibersite, which, while rare in the Earth's crust, is more common in metallic meteorites, and these would have fallen to the surface regularly 4 billion years ago, and thus be washed into hydrothermal pools. When in water, Schreibersite will corrode, releasing reactive phosphorus species such as phosphite, pyrophosphate, and metaphosphate. In lieu of the phosphoanhydride bond on the ATP molecules, early organisms could have reacted exogenous reactive phosphorus species with organic molecules to phosphorylate them. Once the metabolic reaction was completed, the inert orthophosphate would be released and excreted as a waste product, while other reactive phosphorus species would be used to phosphorylate the remaining organic molecules. So, the primordial energy metabolism can be described as dissimilatory energetic phosphate hydrolysis. As a general rule, serpentinizing systems are quite basic, having pHs close to 11. Meanwhile, the atmosphere was about 78% nitrogen, 20% carbon dioxide, and 2% other, and the high concentrations of carbon dioxide would have given both the ocean and rainwater a pH of around 6. As a result, the influx of rain into these pools would have driven changes in both pH and temperature, dropping both. Meanwhile, when the pool dried, the pH and temperature would both increase, giving rise to the aforementioned cycles. While carbon fixation can occur biotically and abiotically, in serpentinizing systems, isotope data can help determine the source. Now, time for a quick lesson in isotope chemistry. Carbon has two stable isotopes, 12 and 13, with 12 being the most common, while 13 makes up 1.1% of all carbon nuclei. The abundance of carbon-13 is given by the delta notation. This is equal to 1 minus the ratio of the isotope in question over the most abundant isotope of a sample divided by the ratio of a standard multiplied by 1000. The standard for carbon is the Pd belemnite. Usually, oxidized compounds are more enriched in the heavier isotope, while reduced ones are depleted in it. Furthermore, biotic processes will usually fractionate isotopes more than abiotic ones. Most organic carbon produced via primary productivity has a delta 13C value between -25 and -30 per mil, while abiotically produced organic carbon has a delta 13C value close to 0. Meanwhile, when organic carbon is produced biotically in a carbon-limited environment, the delta 13C value is between -20 and -5. If we look at graphite from Aquileia, dated to 3.85 billion years ago, we can see delta 13C signatures characteristic of both abiotic and biotic carbon-limited production. This suggests that at this time, life was largely restricted to serpentinizing systems. These are one of the few carbon-limited systems because the basic environment causes carbonates to precipitate out as minerals, but life was also producing much of its own organic carbon from inorganic material. Given how deeply rooted its enzymes are, early organisms probably used the Wood-LJungdahl Pathway to fix carbon by reacting carbon dioxide with molecular hydrogen. While carbon fixation can happen by abiotic mechanisms under biologically relevant conditions, most of the time it happens under pressures and temperatures too high for anything to survive. This suggests that by the mid-Eoarchean, organisms had evolved enzymes comparable to the ones in the present day, allowing them to carry out organic carbon synthesis reactions under conditions they could survive in. However, they would probably still have taken various organic enzyme cofactors, i.e. vitamins, along with inorganic ones, i.e. minerals, which were produced abiotically in the environment. Evolving the ability to synthesize these compounds from either simple organic molecules or inorganic ions in the environment would give any organism who possessed such an ability an advantage, but it would also allow early life to venture from its cradle. As stated before, there are also volcanic hydrothermal vents, and, with the higher mantle temperatures at the time, these would also have been more prevalent than they are now, although the percentage of hydrothermal systems which were serpentinizing would have been higher than it is now. Volcanic vents will also emit hydrogen, which was originally dissolved in the magma, along with carbon dioxide. So, these vents could have also acted as a source of hydrogen for early organisms. Unlike serpentinizing systems, volcanic hydrothermal vents and pools are usually acidic, so carbon dioxide is present in higher concentrations than hydrogen, i.e., they aren't carbon limited. If we look at graphite from Isua, dated to 3.77 billion years ago, we can see that while most organic carbon has a delta 13C value characteristic of biotic carbon-limited production, some of it has a value characteristic of non-carbon limited production. This would suggest that while most organisms were living in serpentinizing systems, some had colonized volcanic hydrothermal environments. While life is often said to be carbon-based, other elements such as nitrogen are key components of biomolecules. On these organic molecules, the nitrogen is in the form of amino groups, and of all the forms of nitrogen which can be found in the environment, ammonia is the easiest to assimilate. In the rocks in Isua, ammonium is abundant, implying it was present in high concentrations in the environment billions of years ago. How it got there is a bit of a mystery. In a manner similar to carbon, diatomic nitrogen can be reacted with molecular hydrogen to form ammonia. However, since the triple bond in the nitrogen molecule is stronger than the double bonds in the carbon dioxide molecule, even higher temperatures and pressures are required for this reaction to proceed. Temperatures and pressures that aren't known to occur in naturally occurring hydrothermal systems. In my opinion, the answer lies in the activity of the young sun. At the time, the sun would emit flares hundreds of times more powerful than modern solar storms. Charged particles from these flares would penetrate deep into the atmosphere and hit nitrogen molecules, producing radicals. These then could be utilized in ammonia synthesizing reactions in hydrothermal systems. Apart from volcanic systems, early organisms could start to colonize the oceans surrounding the islands. To deal with the salinity, these microbes would need membrane-bound pumps to remove salt and hydrogen ions from their cytoplasm. They could have accomplished this in one of two ways. One would be to use the hydrolysis of ATP to power dual-function proton-sodium pumps, with the ATP itself being produced by reacting Schreibersite-derived metaphosphate with ADP. The second way is to use the carbon fixation pathway. Since reacting hydrogen with carbon dioxide to produce organic molecules is exothermic, i.e. energy producing, some of this energy could be used to pump ions. At some point, the latter pumped out enough ions that the direction of the former's reaction was reversed. The influx of ions was used to drive the production of ATP from ADP and orthophosphate, giving rise to the ATP synthase enzyme. Now, life no longer required an input of reactive phosphorus species from meteoric Schreibersite, and instead could replenish its own phosphoanhydride bonds from its intracellular phosphorus pool. Although, to get enough energy to do this, the organisms had to metabolize more carbon than they would assimilate, excreting the excess as acetate and giving rise to acetogenesis. Modern acetogens release 90% of the carbon dioxide they metabolize as acetate, as opposed to assimilating it into their biomass. Every mole of acetate produced will yield 105 kilojoules of energy, meaning 525 kilojoules of energy are required for every mole of carbon assimilated. The conversion of chemical energy into the phosphoanhydride bond in an ATP is about 50% efficient. Since the hydrolysis of a phosphoanhydride bond yields 30.5 kilojoules per mole, a precursor to LUCA would have to hydrolyze 8.5 phosphoanhydride bonds for every carbon atom assimilated. At this point, organisms could colonize vents in the deep ocean with minimal delivery of meteoric material. But not only that, in the latter part of the Eoarchean, the late heavy bombardment waned, and less meteoric material was delivered to the surface. With less Schreibersite falling to the surface, organisms who employ dissimilatory energetic phosphate hydrolysis would have a disadvantage over those with the ATP synthase enzyme. In fact, it seems the ATP synthase enzyme dates back to around the same time that the last universal common ancestor existed, and it may explain why the last common ancestor is the last common ancestor, i.e., its immediate descendants quickly outcompeted those who employed dissimilatory energetic phosphate hydrolysis. Now, I think I should mention something about the late heavy bombardment, which is that some question whether or not it happened at all, the details of which are a topic for another video. But that the Earth was getting hit at high rates around the Hadean-Archean boundary, and this rate declined over the course of the Eoarchean was never in question. What is disputed is whether or not the rate of impact shot up in the late Hadean and declined in the Eoarchean, or whether the decline seen in the Eoarchean was a continuation of a trend that began when the Earth formed. Something else I should address is that recently, some papers have come out arguing that LUCA lived much earlier sometime in the late Hadean. shortly after the surface of the Earth had cooled sufficiently for liquid water to exist. These used molecular clocks, which assume that the mutation rate of the immediate descendants of Leuca were in between that of the common ancestor of bacteria and that of archaea. However, bacteria and archaea use different, i.e., non-homologous enzymes to carry out DNA replication, implying Leuca used ribozymes to do this. These have a greater error rate than enzymes, so, in the branches leading up to bacteria and archaea, the mutation rate would have been even greater. Since the time estimates given by molecular clocks are based off the number of mutations, the rapid accumulation of mutations would make Leuca appear older than it is. If the last universal common ancestor, or Leuca, lived in a deep-sea vent, it would not only not have access to Schreibersite-derived reactive phosphorus species, but not as many nitrogen radicals would enter the system to be converted into ammonia. As such, it probably would have to acquire nitrogen from other sources. Many modern organisms can obtain nitrogen in the form of diatomic nitrogen via the nitrogenase enzyme. Phylogenetic analysis has indicated that while Leuca probably didn't have a functional nitrogenase, it likely had a progenitor to it. This precursor could have instead metabolized hydrogen cyanide, which Leuca used as a nitrogen source. While cyanide is virtually non-existent in modern hydrothermal systems, during the Eoarchean, the mantle had yet to emit all of its nitrogen, and thus hydrogen cyanide would have been common in ancient hydrothermal systems. Other biophilic elements such as sulfur and iron are emitted by hydrothermal vents in bioavailable forms, i.e. as sulfide and ferrous iron. After Leuca, other forms of dissimilatory carbon metabolism could have evolved. For one, biosynthesis pathways could be reversed, giving rise to fermentation, where complex biomolecules are broken down into simple organic ones, such as acetate, to produce energy. Leuca gave rise to the two main divisions of life, one being bacteria and the other being archaea. On the archaeal branch, another new form of dissimilatory carbon metabolism known as methanogenesis evolved. Like acetogens, methanogens use the Wood-LJungdahl Pathway to reduce carbon, but these reduce it all the way to methane. The original form was likely hydrogenotrophic, but via modifications to the pathway, two other forms could have evolved: acetoclastic and methylotrophic methanogenesis. Acetoclastic methanogenesis will split acetate into methane and carbon dioxide. Meanwhile, methylotrophic methanogens metabolize methylated substrates like dimethyl sulfide, methylamine, and methanol, and will reduce most of the methyl groups to methane while oxidizing some to carbon dioxide. Once fermentation began, organisms living in the periphery of the hydrothermal vents could break down organic molecules produced by organisms within it as a means of obtaining energy. Furthermore, once methanogenesis evolved, acetoclastic methanogens could metabolize the acetate produced by hydrogenotrophic acetogens, or that produced by fermenters, although less acetate would be produced because more primary productivity would be carried out by hydrogenotrophic methanogens. While we may think life at this time would be largely confined to hydrothermal systems, recent research has shown that various microorganisms can survive and even grow by oxidizing trace gases, that being hydrogen, methane, and carbon dioxide, which exist as minor constituents of the ambient atmosphere. The role reduced gases such as hydrogen, methane, hydrogen sulfide, and ammonia played in the origin of life have been extensively investigated in the latter part of the 20th century. This is because it was thought that the primordial Earth had a reducing atmosphere, and, as the Miller-Urey experiment demonstrated, passing lightning through such a gaseous mixture will result in the production of simple organic molecules. However, interest in such atmospheric gases waned after analysis of ancient zircons indicated that the primordial atmosphere wasn't reducing but inert. But the revelation that organisms can survive off trace gases in the modern atmosphere does open up some interesting possibilities. Oxidizing all of the trace gases in 1 liter of air will yield 73.4 millijoules of energy. While hydrogen wasn't a major constituent of the Eoarchean atmosphere, it was certainly present in much greater concentrations than today. So, if the Eoarchean atmosphere had a hydrogen concentration greater than 67.5 parts per million, an acetogen could get more energy from it than a modern trace gas oxidizer, and if it was greater than 54.1 ppm, a methanogen could do the same. As such, microorganisms could have colonized the open oceans via surviving off atmospheric carbon dioxide and hydrogen. Of course, life needs more than energy and carbon. It also needs other elements like nitrogen and sulfur. Well, in prokaryotic organisms, the carbon and nitrogen ratio is usually around 4:1. An acetogen will metabolize 20 hydrogen molecules for every carbon atom assimilated, so it would need 1 nitrogen for every 80 hydrogen molecules metabolized. This means that if the atmospheric concentration of hydrogen was 67.5 parts per million, an acetogenic trace gas oxidizer would need an atmospheric ammonia concentration of 844 parts per billion and a hydrogen sulfide concentration of 32 ppb to meet its nitrogen and sulfur demands respectively. Although, I should mention that even if microbes could expand into the open oceans at this time, their population would be so low that it wouldn't leave a visible trace. Regretfully, it's hard to piece together what was going on in the late Eoarchean and early Paleoarchean because there are very few sedimentary rocks from this time. If the Eoarchean could be called the Hydrogen Age, the Paleoarchean would be called the Sulfur Age. Before we discuss this era, we should discuss sulfur isotope chemistry. While carbon has 2 stable isotopes, sulfur has 4, 32, 33, 34, and 36. The most abundant of these is sulfur 32 at 95.02%, followed by sulfur 34 at 4.21%. Next is sulfur 33 at 0.75%, and finally sulfur 36 at 0.02%. Most processes which fractionate sulfur isotopes, including biological metabolism, do so in a manner which is mass-dependent. This means the fractionation of the isotopes is proportional to their difference in mass with sulfur 32. For example, if you have a material with a delta 34S value of 0 and subject it to a process which fractionates sulfur isotopes, the change in the delta 33S value will be equal to the change in the delta 34S value times 0.52, which is known as the mass-dependent fractionation, or MDF slope. If you divide the difference in mass between sulfur 33 and 32 by the difference between sulfur 34 and 32, you get 0.5. The reason it's 0.52 is because of the difference in mass between protons and neutrons. However, processes which don't fractionate isotopes in a manner which land on the MDF slope are known as mass-independent fractionation. How far the delta 33S value is above or below the slope is the capital delta 33S value. It's a similar thing with sulfur 36, but the MDF slope here is 1.9. One process which is known to fractionate sulfur isotopes in a mass-independent manner is UV photolysis. Here, UV photons will strike atmospheric sulfur dioxide, producing elemental sulfur and sulfate, with the elemental sulfur having a positive capital delta 33S value, while the sulfate has a negative value. Throughout the Archean, we see widespread evidence of mass-independent fractionation, but it's not until the Paleoarchean that we find evidence of large-scale mass-dependent fractionation, suggesting that at this time, organisms began metabolizing sulfur. When microbes reduce sulfur, using it as the terminal electron acceptor, the resulting sulfides will have a lower delta 34S value than abiotically produced sulfides. Conversely, when microorganisms oxidize sulfides, they will not fractionate the isotopes at all, while abiotic oxidation processes will enrich the resulting sulfate in sulfur 34. In rocks from 3.5 billion years ago, we see sulfides with very low delta 34S values and sulfate with a delta 34S value close to 0, implying both processes were active. Dissimilatory sulfur metabolism likely begun in the early Paleoarchean, with microorganisms using sulfite as the terminal electron acceptor for the oxidation of hydrogen emitted by the hydrothermal vents as an energy source. The sulfite would have been the result of sulfur dioxide dissolved in the magma reacting with water. These organisms would have also reacted carbon dioxide with hydrogen and used the Wood-LJungdahl Pathway to produce biomass. Once dissimilatory sulfur metabolism had been established, it could expand in one of two ways. First, instead of using hydrogen, a sulfur reducer could use organic carbon as the electron donor, giving rise to the first form of respiration. Second, they could use other oxidized sulfur-based compounds as electron acceptors, such as thiosulfate and sulfate. On the modern Earth, sulfate is produced abiotically when sulfite, produced via reactions between sulfur dioxide and water, react with oxygen. But in the Paleoarchean atmosphere, there was no oxygen. However, sulfite could react with calcium oxide to produce sulfate in hydrothermal systems. In these hydrothermal vents, sulfur reduction would be electron donor limited, since the concentration of sulfur dioxide, and thus sulfite and sulfate, will be greater than hydrogen. You could ask, what about those which used organic carbon as the electron donor? But, since at this point, the amount of organic carbon produced would depend on the quantity of hydrogen, sulfur reduction would still be electron donor limited. Although, since the difference in electronegativity between sulfur and hydrogen is greater than the difference between carbon and hydrogen, more energy can be obtained from each mole of hydrogen metabolized by a sulfur reducer than by a methanogen. This would also mean that more of the hydrogen metabolized could be used for carbon fixation. So, the advent of sulfur reduction would have increased primary productivity, but the genesis of a new type of metabolism would change the face of the Earth forever. This was photosynthesis, but instead of using water as the electron donor, it originally would have used hydrogen sulfide. Evidence of photosynthesis circa 3.5 billion years ago comes in many forms. For one, we find carbonates in what would have been the photic zone with delta 13C values between -25 and -30, characteristic of primary productivity, suggesting it was biomass fixed by phototrophic organisms which was then remineralized. We also find stromatolites linked to hydrothermal systems, such as those in the Strelly Pool Formation in Buck Reef Chert, and gypsum whose sulfate is a delta 34S value close to 0. Instead of oxygen, these early photosynthesizers produce sulfate. If we look at the carbon isotope record again, we can see that after 3.5 billion years ago, most organic carbon has a delta 13C value characteristic of biologically produced molecules. While not as common as they were in the Eoarchean, serpentinizing systems were still abundant in the Paleoarchean, yet very little organic carbon has an isotopic signature of that produced in these environments, implying that what we're seeing is a large increase in the rate of biologically produced carbon, which swamped the signal from abiotically or biotic carbon-limited organic carbon production. However, what we don't see is an abrupt increase in the rate of organic carbon burial in the Paleoarchean. This could be explained by the emergence of respiration, i.e. oxidizing organic carbon as an energy source. In the Eoarchean, organic matter wasn't burned, i.e. oxidized, but broken down by heterotrophic organisms. While fermentation would break down a portion of it into volatile compounds that would escape into the atmosphere, a much greater fraction of the organic carbon produced would be buried. However, once respiration began, most of the organic carbon produced will be oxidized and return to the atmosphere as carbon dioxide. So, while much more organic carbon would have been produced during the Paleoarchean, it would have also been consumed by sulfur aspires. Hence, we see a shift in the isotopic composition of the organic carbon without seeing much of an increase in the organic carbon burial rate. When we again look at the sulfur isotope record, we see some interesting trends. Much of the sulfides from this time period, with delta 34S values characteristic of microbial sulfur reduction, have positive cap delta 33S values. This implies that heterotrophic organisms were using photolytically produced elemental sulfur as an electron acceptor. Now, why is this? Well, as stated before, oxidized sulfur species are more abundant in hydrothermal systems than hydrogen, but hydrogen sulfide is present in greater concentrations than sulfate or sulfite. Now, you might ask, wait, wouldn't these early phototrophs produce sulfate, which heterotrophs could use? Well, yes, but the presence of barium ions in the Archean Ocean would cause it to precipitate out as barite. While microorganisms can use the sulfate bound in barite as their electron acceptor, they can only do so at low rates. This would mean that the advent of photosynthesis caused sulfur respiration to go from being electron donor to electron acceptor limited, which then caused them to utilize elemental sulfur aerosols as an alternative electron acceptor. The sulfide produced by these organisms would then precipitate out as pyrite when it reacted with ferrous iron ions in the water. So, what we have here is a kind of proto-carbon cycle, where carbon is being cycled between phototrophs and heterotrophs, but the electron donors and acceptors are coming from geological and atmospheric sources. We can use the sulfur isotope record not only to identify the presence of UV photolysis, but to identify what varieties of it were active at any given time. When photolytically produced sulfur compounds are deposited and mixed to varying degrees, they fall on lines or arrays when you plot the cap delta 36S and cap delta 33S values. In the Eoarchean, much of the sulfur falls on a broadband array, which is what you get when you expose sulfur dioxide to UV light with a wavelength between 180 and 220 nanometers. Meanwhile, in the Paleoarchean, most of it falls on the 193 nanometer array, and the total mass independent fractionation is less. This would suggest that some kind of UV shielding was present in the Paleoarchean. The most likely candidate is carbonyl sulfide, or OCS, which has a minimum absorbance at 193 nanometers. This compound is produced when carbon monoxide reacts with elemental sulfur. The carbon monoxide itself could be produced by UV light striking various dissolved organic molecules produced by phototrophic organisms. The UV shielding produced by OCS haze would in turn allow for greater rates of photosynthesis, leading to a positive feedback loop. Something I should mention is that chlorophyll and similar molecules like bacteria chlorophylls aren't the only photosynthetic pigments. There are also retinols, which are used by photoheterotrophs, organisms which uptake organic carbon but use light energy to power their metabolism. Some have hypothesized that the first photosynthetic organisms use retinols versus chlorophylls, i.e., the purple earth hypothesis. This has been reinforced by phylogenetic analysis, which has indicated that some of the retinols date back to Leuca, while it doesn't appear that chlorophylls go that far back. Also, if you look at the absorption spectra, you can see that retinal has a peak absorbance similar to the sun's emission. It has been argued by proponents of this hypothesis that these first evolved to maximize the absorbance of light from the sun, while chlorophylls originally evolved their spectra to scavenge the light left over by retinal-using phototrophs. But there is a problem with this hypothesis. Since photoheterotrophs use retinols, they need a source of organic carbon, and this wasn't common on the surface of the Eoarchean Earth. Going back to the absorption spectra, while that of the retinols closely resembles the emission spectra of the sun, water has a minimum absorbance in the blue light range. Chlorophylls and bacteria chlorophylls have an absorbance peak in this range, meaning that while retinols could absorb more light on the surface, if you go a few meters deep, chlorophylls will be able to absorb more of the light energy. This would have been an even bigger advantage in the Archean than it is now, since, due to the lack of an ozone layer, the UV light intensity would be greater in the surface waters, where retinal-employing phototrophs would have the advantage. Since chlorophylls are employed by organisms which can take carbon as carbon dioxide, they could grow in areas with little to no organic carbon present, i.e., they could grow in places where there wouldn't be any retinal-employing phototrophs to compete for light with, making it unlikely that they evolved their spectrum to harvest the leftover light. Instead of using light as an energy source, an alternative explanation is that they used it to relieve their energy demands. Like most other organisms, photoheterotrophs use a cross-membrane gradient to drive their ATP synthase enzyme. This cross-membrane gradient is generated by light-powered pumps which contain the retinals. Going back to when life used assimilatory energetic phosphate hydrolysis, when organisms began to colonize salty and/or acidic environments, they would have to pump ions out of their cytoplasm. This in turn would require them to use some of the reactive phosphorus species uptaken to power these pumps. Having light-powered pumps would relieve the energy demands, allowing more reactive phosphorus species to power other metabolic processes. Also, I should note that there are reasons to believe that while not responsible for primary productivity, retinal-based phototrophy was more prevalent than it is now. Remember that sulfur respiration in the periphery of the hydrothermal systems was electron acceptor limited, i.e., there was a lot of organic carbon, but without an electron acceptor, there's no energy to drive the synthesis of biomolecules to assimilate into the biomass of sulfur respirers. So, close to the vents, you would have had photothiotrophs metabolizing sulfide to drive the production of organic molecules, while photoheterotrophs lived in the periphery, uptaking dissolved organic carbon released by the photothiotrophs. Meanwhile, sulfur reducers oxidized organic carbon which was buried along with the elemental sulfur aerosols. At this time, most phototrophs would have been restricted to the seabed because the influx of UV light would kill any planktonic organisms. Now, a simple calculation would show that even back then, UV light was only really intense in the top meter or so of the water, and plenty of photosynthesis occurs below 1 meter deep. This is true, but the top 20 to 30 meters of the ocean are well mixed by wave action, and most phototrophic planktonic organisms have a generation time close to a day. So, virtually all of them will be in the top 1 meter of the water column, where they'd receive a lethal dose of radiation before they could reproduce. It is true that organisms living on the seabed will be exposed to similar or even greater fluctuations in UV intensity due to the tides. In fact, back then the tides were greater than they are now because the moon was closer to the Earth. But these fluctuations would have occurred over the course of hours versus seconds, giving the organisms time to acclimate. They could do this by going dormant, burrowing into the soil, or producing photoprotective pigments. The advent of photosynthesis would have opened up many microbial niches, and we can see evidence for this if we look at the tree of life. Using molecular clocks, we can infer that the last common ancestor of all living bacteria, or LBCA, lived shortly after Luca, sometime in the Paleoarchean. There have been numerous attempts to determine exactly how the major groups of bacteria are related to each other. One view has a few minor phyla such as Dermatoga, Aquaficiae, and the candidate phylum Radiation, or CPR, as the outgroups from the rest of the bacteria. Another view has the main division between two major groups, Pterobacteria and Graciculites. Of these, Pterobacteria are more conserved, and in this view, it contains the aforementioned outgroups. While there is an ongoing debate as to what the bacterial phylogeny looks like, the fact that it is this difficult to resolve indicates that the major groups of conserved bacteria diversified rapidly shortly after the last bacterial common ancestor lived, i.e. in the Paleoarchean. It may very well be that bacteria underwent an adaptive radiation as a result of the emergence of all the microbial niches which came about from the advent of photosynthesis. So, was the expansion and development of the biosphere in the Paleoarchean the result of photosynthesis originating, or was it also tied to geologic events? When we look at the hafnium isotope record, we can see continental crust starting to appear some 3.5 billion years ago. Continental crust itself is formed when magma flows upward through oceanic crust, and as it cools, the denser elements, i.e. the mafic ones, precipitate out deeper due to their higher melting temperature, while the lighter, i.e. felsic ones, continue to flow up until they cool sufficiently, leaving the resulting rocks enriched in the elements characteristic of continental crust. As the mantle cooled over the course of the early Archean, the crust differentiated from it, going from ultramafic to mafic as it was enriched in lighter elements. By the Paleoarchean, some of the islands started to be formed from continental crust. Some have argued that the Barberton Greenstone Belt in South Africa and the Pilbara Craton in Australia were combined in a protocontinent known as Valbara, but this is still debated. One of the elements present in greater abundance in continental crust is phosphorus, which often limits photosynthesis today. Remember that most microbes require 1 phosphorus for every 106 carbon atoms that they assimilate, and when sulfide is photooxidized, it gives enough electrons to reduce 2 carbon atoms to be assimilated. This means that early phototrophic organisms would require 53 sulfides per every phosphate metabolized, and sulfite to phosphate ratios in volcanic vents are almost always much greater than 53 to 1. So, before the first continental crust, photosynthesis would have been phosphorus limited, but after it came about, weathering of the lands would have fertilized the oceans with phosphorus. As such, it's hard to say at this point whether the expansion of the biosphere was driven by the evolution of a new system, i.e. photosynthesis, or if it had existed before and only became as common as it did as a result of the influx of phosphorus. With regards to the other major biophilic element, nitrogen, it's likely that the nitrogenase enzyme came about around this time, meaning phototrophs could uptake it as diatomic nitrogen. Next, we come to the Mesoarchean, or the Iron Age of metabolism. In this time period, 3.2 to 2.8 billion years ago, banded iron formations become widespread. Now, there are 2 types of banded iron formations, or BIFs, the superior and Algoma type. The superior types are thicker and formed by dissolved ferrous iron chemically reacting with oxygen in the water and precipitating out as rust. Meanwhile, Algoma types are much finer. Some papers have argued that there are whiffs of oxygen in the Mesoarchean oceans, resulting from the limited production of oxygen by the progenitors of cyanobacteria. This was based on the presence of iron and manganese oxides, which seem to have been formed by interactions between manganese 2 and iron 2 and oxygen, as oxygen is the only element present in sufficient quantities on the Earth that can oxidize them. However, one alternative explanation is that phototrophic organisms photooxidized iron and manganese themselves, using the electrons to reduce carbon dioxide, which in turn drove primary productivity. There is evidence for this found in much more recent rocks. Today, there are organisms which use iron as the electron donor in photosynthesis, known as photoferrotrophs. In what would have been an anoxic basin in the Aegean Sea 2 million years ago, we find Algoma-type banded iron formations, along with the microfossils of photoferrotrophs. So, it may be that similar organisms produce the Mesoarchean BIFs. Now, we also find Algoma-type BIFs in the Eoarchean, but whereas those in the Mesoarchean and of 2 million years ago are composed of both magnetite and hematite, the ones from the Eoarchean are composed solely of magnetite. Instead of being formed from iron oxide precipitates, those in the Eoarchean were likely originally laid as alternating layers of iron and silicate clays. Later, geochemical reworking of the rocks, or diagenesis, partially oxidized the iron clays to magnetite. 1/3 of the iron atoms in magnetite are iron 2, while 2/3 are iron 3. Meanwhile, all of the iron atoms in hematite are iron 3. Hence, magnetite is partially oxidized, while hematite is fully oxidized. When we look at BIFs from the Mesoarchean, we can see that the carbonates in the magnetite nodules have lower delta 13C values than those in the hematite nodules. This suggests that iron-respiring organisms were active here in the Mesoarchean, oxidizing organic carbon, using ferric iron as the electron acceptor, producing ferrous iron, which was later incorporated into the magnetite. If this is the case, it would appear that dissimilatory iron reduction had begun around this time. Indeed, if we again look at the sulfur isotope record, we can see a drop in the degree of mass-dependent fractionation of the sulfides, along with a decrease in the abundance of sulfate, a product of phototrophy. With ferrous iron being one of the most abundant ions in the oceans, while sulfides were largely restricted to geothermal sources, photoferrotrophs would have an advantage over photothotrophs. Meanwhile, since the difference in the half-reduction potential between ferrous and ferric iron and organic carbon and carbon dioxide is greater than that between sulfide and sulfate and organic carbon and carbon dioxide, dissimilatory iron reducers could get more energy per gram of organic carbon metabolized than sulfur reducers, and thus outcompete them. So, it appears that in the Mesoarchean, dissimilatory iron metabolism displaced dissimilatory sulfur metabolism as the main force driving the biosphere. At this point, ferrous iron used by the phototrophs would be regenerated not only by volcanic hydrothermal emissions, but by iron-respiring organisms. This would mean that not only were carbon and nutrients being cycled between producers and consumers, but electron donors and acceptors were as well, giving rise to something resembling the modern carbon cycle, except with oxygen being switched out with iron. Although, in the modern oceans, respiring organisms are active in the water column, whereas fermenters are active in the sediment. This is because respiring organisms metabolize all the electron acceptors in the top layer of the sediment. However, in the Mesoarchean, the iron-3 produced by photoferrotrophs would rapidly precipitate out as rust and sink to the bottom. As a result, iron respirers would be most active on the seabed, while fermenters would be the most active in the water column, which would be in stark contrast to today. If we look at the sulfur isotope record again, we can see that not only does mass-dependent fractionation go down, mass-independent fractionation does as well, implying there is more UV shielding than there was in the Paleoarchean. One hypothesis put forth is that a layer of organic molecules produced via photochemical reactions, or tholin haze, formed in the atmosphere. Today, algae, which comprise the phytoplankton, release many volatile organic compounds that diffuse into the atmosphere, but these are oxidized by oxygen. In the Mesoarchean, the abundance of ferrous iron in the oceans would allow primary productivity to proceed at greater rates than before, and photosynthetic bacteria would also release volatile organic compounds. With no oxygen in the atmosphere, these volatile organics would build up and thus form a haze. This haze layer could very well have been better at shielding the surface from UV light than the sulfur isotope record would lead you to believe. While lower than it was in other eras of the Archean, Mesoarchean mass-independent fractionation was greater than it is now. But remember that only about 3% of the mass of the atmosphere is above the ozone layer, while if a tholin haze existed in the Mesoarchean, it would have done so at the tropopause. About 30% of the mass of the atmosphere lies above this altitude, meaning a much greater fraction of the atmosphere will be subjected to UV photolysis than today, while there is similar UV flux on the surface. This shielding could in turn allow photosynthetic microorganisms to colonize the water column, giving rise to the first phytoplankton. This could also explain why chlorophylls have the absorption spectra that they do. Bacteria chlorophylls and chlorophylls have two absorbance peaks, but the bacteria chlorophylls are spaced further apart. Many hypotheses have been put forth as to why this is. Some have argued that the long wavelength peak was to absorb more of the near-infrared light, which compromised a greater portion of the solar emissions than it does now. Others have argued that the short wavelength peak was to absorb more UV light and thus provide the organism with some shielding. I've done simulations on this, and it turns out that there is limited UV shielding, and while bacteria chlorophylls absorb more light from the young sun than chlorophylls, the same is true for the modern sun. The answer to why bacteria chlorophylls and chlorophylls have different absorption spectrums may lie in what's known as photoinactivation. This is where a sudden increase in the light intensity causes the photosystem to blow out. The short wavelength peak in chlorophylls is very close to the absorption minimum of water. This means that blue light intensity, and hence the light absorbed by chlorophylls, will change the least as a planktonic phototroph is moved vertically in the water column. This in turn would reduce the risk of photoinactivation as the organism is moved toward the surface by wave action. Meanwhile, since bacterial chlorophylls were first used in the Paleoarchean on the seabed, the alterations in light levels the organisms which possessed them were exposed to took place over the course of hours as a result of the day-night cycle, preventing rapid changes in light intensity. So, it may be that chlorophylls evolved their absorption spectra as a result of transitioning from a benthic to a planktonic lifestyle. Analysis of oxygen isotopes indicate that the temperature was much warmer in the Mesoarchean than it is now, and this could have come about as a result of an increase in methane production. What caused this? Well, recent research has shown that while most methanogens crumble when exposed to even very mildly oxidizing conditions, in sandy sediments, some methylotrophic methanogens can survive via metabolizing methylated compounds released by algae. This microaero-tolerant methanogenesis could have its origins in the Mesoarchean, where the somewhat oxidizing conditions came out as a result of iron oxides being produced by photoferrotrophs versus oxygen itself. Such organisms could have metabolized methylated compounds produced by ancient photoferrotrophs, releasing the methyl group as methane, inducing a greenhouse effect, and spiking the temperature. Near the end of the Mesoarchean, we find isotopic evidence of methanotrophy, or oxidizing methane as an energy source. Even though dissimilatory sulfur metabolism is less abundant in this time period than it was before, these first methanotrophs use sulfate as their electron acceptor. Now we come to the last era of this eon, the Neoarchean. This period would be described as the oxygen age, in that we see the beginning of oxygenic photosynthesis, which oxidized water, producing oxygen. Now, phototrophic organisms had access to an electron donor that was what the environment they lived in was made of. From this point on, primary productivity would be limited by either the availability of light or nutrients. Because oxygen is more electronegative than either iron or sulfur, respiring organotrophic organisms could get even more energy per gram of organic carbon metabolized. However, phylogenetic analysis of the genes which reduce oxygen indicate that they originally diversified in iron-oxidizing bacteria. Given the abundance of ferrous iron in the oceans, reacting them with oxygen would be a readily available means of producing energy. Meanwhile, the abiotic oxidation of iron would result in the production of the aforementioned superior type BIFs, which are abundant in Neoarchean age rocks. Meanwhile, near hydrothermal systems, sulfur oxidizing organisms would use the oxygen to oxidize the sulfides to sulfate. These could get far more energy per mole of oxygen and sulfur metabolized because of the greater difference in reduction potential between sulfate and sulfide and oxygen and water. Around this time, sulfur oxidizers evolved the capacity to oxidize other forms of sulfur. In Neoarchean aged rocks, we find sulfate with a positive cap delta 33S value. This implies that it was originally photolytically produced elemental sulfur, which was later oxidized upon deposition. Going back to the sulfur isotope record, we can see that mass-independent fractionation has a cap delta 36 to 33 ratio similar to that of the Eoarchean, i.e. the broadband array, implying any kind of shielding haze layer was removed. What's also notable is that the cap delta 33S values are much greater than in previous eras. This has perplexed geochemists because no photolytic process has been known to produce a cap delta 33 value as high as those seen in the Neoarchean. However, what's different about the Neoarchean was that there was atmospheric oxygen, i.e., A photolytically produced elemental sulfur molecule could react with oxygen, and thus be converted back into sulfur dioxide. Since this chemical reaction results in mass-dependent fractionation, the resulting sulfur dioxide has a positive cap delta 33S value. So, it could undergo a second round of photolysis to produce elemental sulfur with an even higher cap delta 33S value. The sulfate produced by the initial reaction wouldn't be reacted via any atmospheric processes, explaining why Neoarchean mass-independent fractionation values become very positive, but not very negative. The return of intense UV light to the surface would restrict phototrophy to the benthos again. Indeed, we see many photosynthetic microbial mats or stromatolites in nearshore deposits from the Neoarchean. These have rare earth element abundances and isotopic signatures characteristic of environments with oxygen production. Analysis of iron isotopes indicate that both iron oxidizers and iron reducers were active in proximity to the stromatolites. Although oxygen was present, it wasn't produced at sufficient rates to build up in the atmosphere. This could be either because of the lack of UV shielding, which inhibited photosynthesis, or due to the fact that these early cyanobacteria had yet to evolve the thylakoid membrane, or a combination of the two. It appears that by this time, both nitrification, or oxidizing ammonia using oxygen as the electron acceptor to produce nitrate, and denitrification, or oxidizing organic carbon using nitrate as the electron acceptor producing diatomic nitrogen had evolved. But, while most of the inorganic nitrogen in the modern oceans is in the form of nitrate, in the Neoarchean it was in the form of ammonia, indicating that most of the water column was more reducing than today. Throughout the Archean, rates of volcanism steadily decreased as the mantle cooled, yet in the Neoarchean, there is much more volcanism than there is today. Hydrothermal vents would have released copious amounts of reduced iron and sulfur into the oceans. So, while most oxygen produced today is consumed by organotrophs who use it to oxidize carbon, at this time, most of it was probably used by lithotrophs to oxidize these elements. In the Neoarchean, some microorganisms who evolved in the oceans began to recolonize freshwater systems and even the land. For one, we find fossil stromatolites in what would have been lake beds. Furthermore, it seems some endolithic organisms had colonized the land by the early Neoarchean. These organisms live within rocks which could have protected them from the harsh UV light, and these microorganisms could have facilitated continental weathering, washing more nutrients into the oceans. This would in turn fertilize them, but it would also bring in more reduced compounds, which would have kept much of the water column reducing. Continental crust formation, which had begun in the Paleoarchean, continued at greater and greater rates throughout the Mesoarchean, and this rate peaked in the early Neoarchean. It's been proposed that the assorted pieces of continental crust assembled into the quote-unquote supercontinent of Keinorland some 2.7 billion years ago. This continent would have been of comparable size to Eurasia and represented the start of the supercontinent cycle, which saw the formation of the later supercontinents of Columbia, Rodinia, and most recently Pangaea. If we look at the carbon isotopic record, we can see that a lot of the organic carbon produced has a delta 13C value much lower than most biotic primary productivity processes result in. One pathway that fractionates carbon isotopes this much is the Wood-LJungdahl Pathway, employed by the aforementioned methanogens and acetogens. Now, biomass produced via this process has a delta 13C value similar to that produced by other metabolisms, with the lighter carbon being released as methane and acetate. Methane, being more volatile, will be released into the atmosphere, and this atmospheric methane will have a delta 13C value lower than most biomass. Near the end of the Mesoarchean, we see the emergence of the first methanotrophs, and this metabolism would certainly have expanded throughout the early Neoarchean. While methanotrophs oxidize methane as an energy source, They will also use it as their sole carbon source to produce biomass. This means the delta 13C value of methanotrophs will be much lower than most other biomass. While intuition would state that the methanotrophs had simply used up all the methane in the atmosphere, we see organic carbon anomalously depleted in carbon-13 throughout the Neoarchean. Had we seen a sudden drop in the delta 13C value at the start of the Neoarchean, it would suggest that the methanotrophs had simply used up the atmospheric reservoir, but the pattern we see suggests continuous methane production. Even though oxygen is plentiful in the modern atmosphere, sulfate reduction is responsible for about half the organic carbon oxidation in all the oceans. This is largely because sulfate is the third most abundant ion in the oceans. Although sulfate was not as prevalent in the Archean oceans, ferrous iron was fairly common, but the oxidized form of it, which can be used as an electron acceptor, precipitates out as rust, i.e., it would not have been as readily available to anaerobic respirers then as sulfate is now. Near the end of the Neoarchean, weathering of the continents washed more sulfur into the oceans, making it more abundant than iron, but the relative scarcity of electron acceptors in the Neoarchean implies fermentation was more prevalent than it is in the modern oceans. This would also mean that quite a large portion of the organic carbon metabolized will be released as methane. So, unlike today, methane would have been quite a large cog in the carbon cycle. Organic carbon produced by phototrophs would be consumed by heterotrophs, releasing most of it as carbon dioxide, but some as methane. Meanwhile, some of the quote-unquote primary productivity will take the methane from the heterotrophs and the oxygen from the phototrophs to produce its own biomass. The advent of oxygenic photosynthesis would have opened up many new niches, and we can see evidence for this if we again look at the bacterial family tree. The other major group of bacteria, Graciculites, seems to have diversified around this time, taking advantage of the new niches in the environment. So, just as the advent of photosynthesis caused the initial adaptive radiation of bacteria, the emergence of the modern form of it caused the second great radiation. In this radiation, we see the ubiquitous proteobacteria emerge, some of whom became our mitochondria, which in turn allowed for the evolution of eukaryotic and later multicellular life. The Archean eon gave way to the Proterozoic circa 2.5 billion years ago with the Huronian glaciation. It's often said that for the first 85% of the history of life on Earth, all we had was simple microorganisms. But this perspective falsely leads one to believe the world was static and unchanging, just being covered in slime until about 620 million years ago. It also sells short the metabolic diversity of prokaryotic organisms. While eukaryotic organisms have far more morphological diversity, at the biochemical level, all they can do is oxidize organic carbon using oxygen as the electron acceptor, and of course some can photosynthesize. Meanwhile, bacteria have so many exotic dissimilatory metabolic processes, using all kinds of carbon sources and energy sources. These can be in the form of light or redox chemistry, while utilizing so many electron donors and acceptors. How these systems evolve from something like an acetogen, or even something more primordial, into the oxygen-using and producing metabolisms that drive the biosphere today over the course of the Archean is every bit as fascinating as how animals evolved from the Cambrian explosion to what we have today over the course of the Phanerozoic. Many have stated that if we were to look for life on other planets, all we'd find are microbes. While the thought of this may be disappointing to a lot of people, once you truly appreciate how diverse microorganisms are and how dynamic the Archean, or the age of prokaryotes, truly was, this becomes a truly exciting possibility.